Oh, I thought you were going to say
I don't
something.
have my gang.
You don't have your gang.
It's
All
not the
right,
same.
so we're going to be in box number 3. And we're going to keep talking about meiosis today. So we've talked about prophase 1 and metaphase 1, and next is going to come anaphase 1. So I'm going to start at the top by drawing my cell. Now, what happens in anaphase of mitosis? What splits apart? The chromosomes get split apart. The only difference here is that instead of lining up straight up and down, how did the chromosomes line up? They lined up in pairs. And what did we call those pairs? Homologous. We call it a tetrad, or homologous, meaning that the one from dad that codes for the same traits as the one from mom, they get together. So I'm going to use some colors because I'm special and I can. But basically, they're going to get pulled apart in anaphase 1. So what's this fiber that comes out and grabs onto them? The spindles. And since this is an animal cell, it's connected to the centrioles. We can label these spindle. We're going to call this anaphase 1. Now again, why do we have to call it anaphase 1?
Because it's not meiosis.
It's meiosis and not mitosis. And it's also, there's going to be an anaphase 2, right? So if I want to put this into words, I can say homologous pairs, homologous chromosome pairs separate and head to the poles. Now, how many chromosomes did we start out with in our cell?
4.
We start out with 4. But you'll notice when this cell division is done, we're going to have 2 chromosomes in each cell. Now, yeah, they'll be replicated, but still, we really only have 2 chromosomes in each cell. So we're cutting the number of chromosomes in half. So, anaphase 1. All right, what's going to come after anaphase?
Telophase 1.
Telophase 1, and what do we call it when the cytoplasm splits? Cytokinesis.
Is it 1?
Well, they happen together simultaneously. No. We don't. We just call it cytokinesis. We don't call it cytokinesis 1. Okay, so what I'm going to do is now we're going to draw telophase 1 and cytokinesis. Telophase 1 and cytokinesis. And so I'm going to draw 2 cells that are kind of starting to divide. Now these are animal cells, so they're pinching together in a what? What do we call that?
Cleavage furrow.
Cleavage furrow. And what are some things that typically happen in telophase and mitosis? What's something that comes back?
The nucleus.
All right, the nucleus starts to come back, so I'm going to draw the nuclear envelope coming back. How many chromosomes are going to be in each nucleus?
2.
2. So I'm going to draw them. And then some major things here. The nucleus returns. Here's the main thing, though. The 2 daughter cells, they have half the number of chromosomes, so should we call them diploid or haploid? Haploid. Remember, we think haploid means half. So 2 daughter cells are haploid. So we would write that as N equals 2. So our diploid number is what?
2 equals 4.
2n equals 4. So we have cut the number of chromosomes in half. But there's a problem. Do what? Because we started with 4. We started with 4. If you look back at the beginning, we started with 4. And then in meiosis, we're cutting the number in half. but there is one problem.
Uh-huh.
These chromosomes are still replicated, right? In other words, if we look at just this chromosome, it still has two identical sister chromatids. We have two sets of DNA here. And we don't want that. We want one set of DNA in each cell, correct? So what we've got to do is we've got to do another round of cell division to split these up. So that's going to be meiosis 2.
Do we have to like draw two cells and show both of them doing it at the same time, or just draw one?
We're going to draw both cells when we get down here. Because the whole point is when we're done, we're going to have 4 cells. We started, in meiosis, we start with 1 and we end with 4. In mitosis, we start with 1 and end with 2. All right, so down here in box number 5, we're now going to go through meiosis 2, which is a second round of cell division. But now I have how many cells?
2.
I have 2 cells. So I'm going to draw 2 cells.
Do I need to hash this box?
No. I'm going to draw 2 cells. And,
Oh,
yes,
was Carolyn Miles in her first period class?
yes.
Okay, thank you.
Okay.
She finished. You don't even know who she is, do she?
Yeah, she was here. I remember now. Okay, but each of these cells has how many chromosomes? 2. So just like in regular prophase, the chromosomes are going to show up. The centrioles are going to start to develop spindle fibers. Just like you would normally expect in the first stage, which is always what? What do we call the first stage? Prophase, but it's not, what is it? It's prophase what? Prophase 2, not 3. Prophase 2. So we can say that it's same as mitosis. The only difference is we're starting with 2 haploid cells instead of 1 diploid. Now, do we need to replicate DNA? No, because look, we've already got 2 sets of DNA. DNA is already replicated. What are some other things that usually happen in prophase? I've just drawn the chromosomes here and the spindles, but what else happens there? What thing? The nucleolus disappears and the nuclear envelope. So basically the nucleus goes away, and those happen as well. That's why we put it as same as mitosis. And after prophase 2 is going to come metaphase 2.
Race.
Oh my goodness. Okay, so now let's go over to block number 6. And I'm going to draw these cells again. I'm going to draw them a little bigger because I may need a little more room. And this is going to be metaphase 2. What always happens in metaphase?
They line up.
They line up. And they're going to line up just like in mitosis. They're going to line up straight up and down. Just like this. And what attaches to them? The spindle fibers. And these spindle fibers, they do lots of other things. They don't just pull the chromosomes. They actually help push the cell apart to help the cell divide. They have other functions. But for our concern, they're just grabbing those chromosomes. So chromosomes line up at equator just like in what process? Mitosis. Like mitosis. Not your tosis. Mitosis. And if we wanted to label, we could say these are our spindles and these are our chromosomes. So if I were to ask you, say on a quiz or something, what is the major difference between metaphase 1 and metaphase 2? What would you tell me? One person. Think about it and raise your hand. You got it?
In metaphase 1, the daddy cell and mama cell, like they pair up together in the middle.
Not cells, but chromosomes.
Yeah,
Right?
chromosomes.
Okay, keep going.
They pair up in the middle, and in metaphase 2, like they're like already separated, and they just line up like in mitosis.
Right. The main thing is in metaphase 1, they line up as pairs, as homologous pairs. And in metaphase 2, they line up straight up and down, just like in mitosis.
But why?
Correct. So we're going to draw our two cells again, but this time they're in anaphase. And so we pulled those sister chromatids apart, but we don't call them chromatids anymore. What do we call them? Chromosomes. And this is not anaphase, but it's anaphase what?
2.
Anaphase 2. Anaphase dose. And the sister chromatids separate and move where?
Part from where
To the what?
pole.
To the poles. And that's really all that happens there, just like in mitosis. Yes.
Can you fix that?
Fix what?
Meiosis.
Meiosis.
This is Meiosis.
Oh, it does. Meiosis. There should be an I there. There we go.
It was bothering me.
Meiosis. We invented a new type of cell division today.
Yes. We're really smart.
Now, after telophase, or excuse me, after anaphase is going to come Telophase 2 and cytokinesis. Okay, the last step here is telophase and cytokinesis. Right? Don't we call it telophase? Telophase 2. Thank you for being the only person who's paying attention.
2.
Telophase 2 and cytokinesis
0.
is our last step here. So I'm going to draw both of these sets of cells, pinching in half. Now the nucleus obviously is going to come back, but how many chromosomes are going to be in each cell? How many chromosomes are going to be in each cell?
1,
1.
2. There's going to be 2. Look at what happens in anaphase. There's 2 going each side. So I'm going to draw 2 chromosomes in each cell. So these cells split, resulting in how many cells total?
4.
Resulting in 4 haploid cells. If it's male, we would call these cells what? Sperm. If it's female, we will call them eggs. Four haploid daughter cells.
Okay, now can you explain the math, how you got the little half, 2 halves